Okay, well, here we are. One of my favorite and frequent guests on Healthcare Unfiltered. I'm always grateful to have Vincent Rajkumar on the Healthcare Unfiltered podcast because I know how busy you are, Vincent, and taking time of your busy schedule is always a privilege and an honor. So thank you so much. But I wanted to introduce yourself to listeners. The one commentary I have is we're not talking anything COVID or pandemic, which is more fun to talk about, go back and talking about trials and science and oncology and other things. So, few folks out there don't know you and don't know what you do. Tell us a little bit about you. And I'll have to say, actually, I want to put a link in the podcast notes to one of my favorite, I think it was a Lancet article, talked about your journey and how you got where you are. I think a lot of folks will enjoy reading this. All right, free flow. Tell us who you are.
Well, Chaddy, thanks so much for having me. Always a pleasure to talk to you. I love the Healthcare Unfiltered podcast and the amazing guests you have and the topics you cover. Um, I work in the myeloma field at Mayo Clinic, and I've been here for more than 25 years. And I do primarily research on myeloma, and I cover clinical trials, epidemiologic studies, as well as laboratory studies. And I've been funded throughout by the NIH. And I also chair the myeloma committee for the Eastern Cooperative Oncology Group. And I edit the Blood Cancer Journal along with Ayalu Teferi.
And then you have what's your involvement, Vincent, with the International Myeloma Foundation and there are a couple of foundations that are pretty broad, and I always see you involved with them. How did this come about, and what's your involvement with them?
So I'm also on the board of directors of the International Myeloma Foundation, and I co-chair the International Myeloma Working Group with Brian Dury. So that has about 250 to 300 myeloma researchers from around the world. And we do both consensus statements and guidelines in terms of whether how is myeloma defined, how is it staged, what's the response criteria that's used, to also research projects where we can bring people from around the world together to work on a question. So that's been a privilege and an honor to do that. And I've been doing that for a long time now.
And today's topic, Vincent, is we're going to talk a little bit in general about randomized controlled trials. The good, bad, and ugly. But before I do that, I'm a little bit disappointed in the training programs where I really don't feel fellows, maybe things have changed since I was a fellow, but fellows never really got primers in designing clinical trials, in the nuances of clinical trials, at least when I was a fellow, which was more than 2 decades ago. But do you feel that fellows in training currently, when they finish their fellowship, are they able to understand the design, the nuances, the pitfalls, the shortcomings, or do you feel we could do a better job in training them and teaching them?
So, I don't know how I can comment on that, having not been involved with many fellowship programs other than the one at Mayo. I trained at Mayo, and I think everything depends on how the fellowship program is structured. Mayo has a very clinically oriented program, and it's also longer than the other programs. It's a 4-year program, and I did all 4 years. And by the time I was done with my fellowship, I had published several clinical trials, three clinical trials. I had started trials that I didn't finish, someone else finished, but I got a very good understanding of how to design a trial, how to run a trial, how to get one approved, so that I finished fellowship in 1999. And in 2000, I was leading a randomized controlled trial for ECOG, which led eventually to the approval of thalidomide. So that's just 1 year out of fellowship. So I owe a lot to the amount of stuff I learned during fellowship. I think to be a successful clinical trialist, you have to start very early because it takes a long, long time. And it depends on the fellowship program. And I do agree with you that fellowship programs should offer the opportunity for fellows to design um, and run trials. Now, things are complicated now because it takes a much longer time to get a trial up and running. The approval process is longer. So a fellow who starts a trial right now is very unlikely to see it finished unless they stay on staff at the same institution.
Yeah.
So somebody else is going to take the credit,
Yeah.
but they would at least get the training on how to write and get a trial up and running.
And who would you consider your mentors? Is Bob Kyle? Are these folks? I mean, the giants in myeloma back in the day, or?
Yeah, when I trained at Mayo, Bob was very much active and was very influential in my career and probably my primary mentor. Bob, however, didn't do that many clinical trials at the time I was training. So I did a lot of what I gained from him was all of the epidemiologic studies, the racial disparities, the prevalence of MGUS, the progression of MGUS, the biomarkers. For clinical trials per se, I think the people who really inspired me and helped me get the trials up and running were partly in the solid tumor field. I did a lot of trials in brain tumors before I went on to be a myeloma investigator. So Jan Buckner, who later on became the head of the NCCTG, he was my primary mentor in terms of how to design and run a clinical trial. I did like 4 or 5 brain tumor trials with him. Another person was Tom Witzig, who I did a couple of phase 2 and phase 3 trials with, and Maury Gertz. I think these people were very influential in helping me, understand what is the process for doing a clinical trial and getting it successfully completed and published.
I did not know that about brain tumors in you. This is a new thing I learned about you today. So let's start by randomized controlled trials. My first question to you is looking at the type of randomized controlled trials, and then is getting funding dependent on the type of randomized control? When you look at randomized control trial, is it just one way, like experimental arm, control arm, and you try to search for somebody to pay money for the experimental arm? Take us through the process and the type of studies we're talking about.
So therapeutic randomized controlled trials, I mean, you can classify them in many ways, but the scientific way is to see if it was a single blind or a double blind. Is it an open label or a placebo controlled trial? Those things are technically important. Then there is the way of looking at randomized controlled trials where you see what is the funding source. Randomized controlled trials today cost a lot of money, and most of the randomized controlled trials in oncology are funded by pharma. And those can come in 2 categories. One is pharma funded and pharma run trials, where they are the stat center. And then investigator initiated trials where pharma is providing funding. So the pharma funded trials are a big chunk of the RCT portfolio. And then there's public funding. In the US, it'll be NIH funded trials, usually run through cooperative groups. And then there might be smaller trials which are single institution or philanthropy funded, but those are very small proportions. So you can look at randomized trials basically based on the funding source. You can also look at randomized trials in terms of what is the purpose of this trial, which is usually very different than what is the primary aim and the secondary aim. But those are technical things. But the purpose of the trial, which is usually not written out in a protocol, is basically, why are we doing this trial? Are we trying to get a new drug approved? Are we trying to get a new indication for an existing drug? Are we trying to find what is the best treatment, like comparing 2 different treatments? both of which are on the market? Or are we trying to make a strategic difference? Like, should myeloma therapy be indefinite, or can we stop it and see how it goes? Or should we do it through MRD directed therapy, or should we not? Should we try to cure it or just control it? Those are strategic questions. So you can look at randomized trials based on what is the purpose of this trial, which unfortunately is not well known except for the PI and whoever the funder is. So I think I think it's it's a complicated um, set of uh, topics here, and depending on how you look at it, things would be different. I am of the firm view that randomized controlled trials are the gold standard and are critical for most important decisions. But because of the time it takes, the cost, the um, the various barriers we have, the limitations of them, and the number of questions we need to answer in clinical medicine versus the number of how much of time and money we have to pick and choose where we do one and where we won't be able to do it. That's just the reality.
Yeah, I've said that actually many times, that I wish we can answer everything with a randomized controlled trial, but we can't. In fact, I could tell you, Vincent, that it's part of the reason I became a fan of real world evidence and registry data, and despite their limitations, but I really feel that you can, in addition to finding some hypothesis generating questions, you can sometimes answer certain questions through real world data without RCTs, but you have to be very selective. You can't answer everything. So I like how you framed it. We can separate clinical trials, randomized control trials, based on the funding source. Okay, so let's start with first funding source. You said sometimes the government funds clinical trial. Are there specific trials that the government would fund, and how easy is that?
So, and again, this is why it's important to know the process and things involved. I've been fortunate that I have been PI of industry-funded, industry-run trial, randomized trial, industry-funded but investigator-initiated trial, NIH funded through my R01 grant, just straight through NIH to Mayo to me, as well as cooperative group trials where NIH funds the ECOG and then ECOG funds the trial.
That's perfect.
So,
take us through each one.
So, and the process is quite different. If, for example, I'll give you real world examples. Okay, so ixazomib, it's a new proteasome inhibitor. I worked with Takeda on the design of the trial, how to do it, and so I was PI of the ixazomib double-blind randomized controlled trials for maintenance and for frontline therapy, and both of those have been published. But there, pharma is the data center. Pharma funds these trials. These trials are extraordinarily expensive because they are regulatory motive. They have to go to the FDA. So you cannot just audit the trial to make sure everything is okay. You have to monitor the trial. Every patient, that's why all these monitors come multiple times to each center to actually see everything is going correctly according to the way the protocol says. The PI's role there is not as hard because I don't have to fight to get it approved. Because as long as pharma and I agree on the design, the trial's up and running because the funder is in place, and we just have to make sure that the FDA is okay with the design, and then you're up and running. And you recruit fast because pharma has more dollars, so you can open the trial worldwide. You can open and close within a few months. So I've done that. There are trials where you say, I have this idea, but I don't think any pharmaceutical company is interested. Plus, I want to do it on my own. So I wrote an R01 grant. So there I have to first tell the NIH reviewers, I would like to do this trial and you need to fund me. And that's usually revolving around a concept. Like, should we treat smoldering myeloma or not? So thalidomide versus nothing. So that's the trial I did, Zometa versus Zometa. The Zometa was just for, bone strengthening, but it was really to see whether thalidomide was useful in smoldering myeloma. And the hypothesis was early therapy. So you write the R01 grant, you get it approved, you have to get a good score, you get the funding. Then you go to Mayo and say, I need IRB approval for this phase 3 trial that I've written, and here's the money for it that I got from NIH. Because without money, they won't let me open. I need a statistician, I need the monitors, I need, make sure that the trial goes on for 10 years. Who's going to pay for all of that? The lab studies, everything. So that's another way of doing it, but that's incredibly hard. I would say the NIH funds very, very few randomized controlled trials through R01 grants. And then
do they usually fund more basic science stuff, R01s?
They usually fund basic science, translational, even clinical trials, phase 1 and 2, but a randomized trial that NIH actually funds to run, I think it'll be very rare. I was lucky to do it, but in those days it didn't cost as much. I don't think I'll be able to get that kind of budget now. And then finally, the cooperative group trials where there's a certain amount of money that NIH has to do important trials that drug companies won't do. Like drug companies will do, say, DARA-RD versus RD, but drug companies won't do a VRD versus KRD because both companies think their regimens are good. Those are the studies only cooperative groups would do. And for that, the process is very hard because there are 4 cooperative groups in the US. And we have a steering committee for each disease group. So if I have an idea for a phase 3 trial, I have to first get it approved through ECOG to make sure that it fits in with ECOG's priorities. Then we go to the myeloma steering committee where members of the other groups are there and other investigators outside of the cooperative groups. And they have to like it and they have to agree with the design. Then it goes to CTEP, who has to see whether of all the trials we should do, is this really one that we are going to spend money on? And then they may like it or not. And then if they like it, then we open the trial, and then you have to be lucky it accrues, because we have open trials where, months go by and nobody's accruing.
Where do you get the funding for that, though, once you open it?
NIH is giving ECOG the money and allowing ECOG to decide, which of the many, many trials should you pursue. So what happens with each of these is that if I were to grade it, on the one hand is the pharma funded trial, and then on the other side is the NIH funded trial. The pharma funded trial funding is plenty. The approval process is easy because there's very few people who can have veto say. And the problem is pharma will control the design. Pharma will get the stats. And they have a real reason why they're doing the trial, usually to get a drug on the market or to get a new drug approval, new indication for an existing drug. NIH trials, you can design whatever you think is the best question that you want to ask for the field. Doesn't have to be to get a new drug approved. It might be a strategic question, a tactical question for the disease. But the funding is limited and you're competing with so many other questions for a small pot of money. And your approval process is much more trickier because you have so many people who are looking at many, many other competing priorities, and they have to decide whether yours is the right one.
So, but even in the first one, so the pharma is sponsoring the trial and they're funding the money and they are integrated in the design. I mean, if you think the design is flawed, you're the myeloma expert, and if every myeloma expert, says, yeah, that design is flawed, you can push back, no? I'm sure this has happened before where somebody probably proposed a design to you and he said, well, no, I don't think that's right.
Of course. And that's where the whole role of trying to understand what is the question that we are trying to answer comes in. And what might look to an outsider like a design that was not right may look to the investigators and the pharma company as a perfectly appropriate design for the situation we find ourselves in. And unless you are privy to those, it'll be very hard for you to understand what is it, why did you do this design? I'll give you an example. We all knew pomalidomide was effective, but pomalidomide, to get pomalidomide approved and available to US patients, we needed to get a randomized controlled trial up and running. And the FDA is ready to approve the drug on a phase 2, but they do need a phase 3 design to go with it. And on the other hand, EU, they want the full trial to be phase 3. So with those questions in mind, investigators have to decide, like, what would be the design that will get us the drug approved fastest? Because none of the investigators were really doubting that the drug worked. So if you were to do a very complicated design, you know that you'll push the drug approval by another 2 years. So for that 2 years, no one's going to have access to that drug. That's the other side that we have to always worry about. And same thing with DARA, same thing with carfilzomib or bortezomib. All of these drugs were approved on phase 2. But that makes the drug available only to Americans. So how do we, when I know DARA works, we can save lives, bortezomib works, how do we as investigators make sure that everybody else around the world has access to the drug? So we pick designs that sometimes for other people may not look correct, but it is actually correct for the purpose that we are trying to do. But your question is, if the design is truly flawed, like I think it is flawed, I will push back. And we have pushed back and said, like, no, we are not going to do that. And they have almost always, the companies are okay with that. When we have all pushed back, many instances where I've been in meetings where together, collectively, the myeloma community has basically vetoed a particular design.
So when you say that some of these newer drugs were approved based on phase 2s, these are the accelerated approval pathway?
Yep.
Now, in the accelerated approval pathway, usually you're supposed to have some post-marketing studies to confirm the findings in the accelerated pathway. Are you usually involved in this as well? After the drug is approved, do you participate in the post-marketing studies, or?
Yeah. And in fact, it's not just post-marketing studies. You have to actually do the phase 3. Yeah. So for example, let's take daratumumab. Okay, when you have patients with relapsed refractory myeloma who have failed everything, bortezomib doesn't work, lenalidomide doesn't work, dex, thalidomide, alkylators, nothing works. They are basically palliative supportive care mode. In that setting, when you give DARA and you find in a phase 1 and a phase 2 trial that a third of the patients are getting a very nice response, and then their symptoms are getting better, and then they are going out for a year or 2 without any progression, what do you as investigator think? You think, wow, we have a really active drug. This drug needs to be available to patients. We can verify if it really works or not, but the pretest probability is very high that this drug actually works. Globally, the situation is, other than the FDA, most other countries will not agree with us. So in the EU or in Asia or any of the other countries, they won't say we'll get data on the market based on your small 100-patient US study. Mm-hmm. Okay? Mm-hmm. They will need a proper phase 3 trial. Show us improvement in PFS or survival in a proper phase 3 trial. In the US, the FDA, recognizing that we could ask for a proper phase 3 trial, but that means for the next 2 years, patients with myeloma will continue to die without having access to data. And so between approving the drug and then finding out if what we did was correct 2 years from now, is that the best thing? Or shall we not approve it and wait for 2 years till they finish their phase 3? That's the dilemma that the FDA faces.
Yeah.
And what the FDA decides is that based on how strong the phase 2 data is, so it's not just post-marketing studies. It's you are supposed to do an actual phase 3 trial and show the FDA that what they approved on an accelerated approval does indeed work. So if I give you the example of daratumumab was approved in the US based on a single-arm phase 2 trial. And the reason it was approved was because in patients who had no other alternatives, it worked a third of the time with a 50% drop in the paraprotein, and patients stayed in good remission for a long period of time. So most of us in the myeloma field were convinced that this drug actually works. There was really no doubt in our mind. So from the FDA standpoint, what they could do is they have two choices. They can go ahead and approve the drug based on what we as the investigators are telling them and based on what we are demonstrating in the phase 2, in which case patients have access to the drug today. Or they can say, you show us the randomized phase 3. If they choose show us the randomized phase 3, then for 2 years or 3 years, patients with myeloma won't have access to that treatment. And there would be many patients losing their lives who could have been saved with DARA. So this is the dilemma. In the US, the FDA is happy with the accelerated approval pathway, and then we'll show the phase 3 results subsequently. In the EU and in other countries, in Asia and others, they are not going to approve a drug based on phase 2. They need the phase 3. So when the drug company faces this problem, what they have to decide is we need the phase 3 to satisfy the FDA requirement. We need the phase 3 to get the drug on the market in Europe and Asia. So you design the phase 3s, and that's what happened with Pollux and Castor. And when the phase 3s come, the drugs get approved in the rest of the world. And the FDA also gives a full approval because whatever they did on accelerated approval is confirmed. Now it goes back to type 1 and type 2 error. The FDA has done accelerated approval for myeloma many, many times. Lenalidomide, pomalidomide, bortezomib, daratumumab, carfilzomib, all of these were approved accelerated approval. All of them, the phase 3 subsequently showed that what they did was the right decision. And because they did the accelerated approval, patients could have access to those treatments for years before people in Europe and other countries could. So that's all very good. But when you do that, there are chances that you might approve a drug that later on doesn't pan out, like it happened with melflufen. That's a risk that we are willing to take.
Yeah, I think that's you just literally said the statement I was going to say. You stole this from my mouth, which is I think that by accelerating the approvals, you do risk the possibility of approving drugs that they may not be as effective as you thought they were. And in fact, they could hurt people. They could hurt patients. So tell me, I mean, when you say that's a risk that we are willing to take, is it because it's going to help advance the field? Is it because pre-approval probability based on preclinical data? Based on how is it? Like, is there a scenario where you say that's a risk we are not willing to take in the accelerated approval pathway versus a risk that you're willing to take? And is there a, I mean, I'm not the patient, you're not the patient. So how do we incorporate patients' voice in this? Because they're really the ones taking the risk. You're not taking it and I'm not taking it.
Absolutely. So it really depends on the disease type and the type of endpoints that you think are reliable. So with myeloma, we are pretty sure that if you have a M protein that goes from 3 grams to 0, it has to do that only with an active drug. It cannot do that with an inactive drug. Okay. So we are sure of the biomarker, we are sure of that. And we're also sure that 9 out of 10 times that patients who respond are going to do better than if you didn't get a response. So in this field, for the type of track record and pretest probability, this is a good pathway. Even then, we've had mistakes happen with melflufen. We've had mistakes happen with where, it didn't get approved, but, we had the venetoclax situation where PFS was longer, but overall survival was not. So mistakes can happen, and we are aware of that. It may not be the same in solid tumors. If you have a new drug and you really don't have a solid pretest probability as on what to make out of the response, or the depth of response, or the extent of the response, you cannot be as confident as I am with myeloma. So I really think it depends on the investigators, and this is why it's important for both pharma and the regulators to listen to the investigators in terms of what we are seeing on phase 2. Is this believable? Will it likely translate to a success on phase 3?
Vincent, one of the most puzzling things to me, and I'm sure to a lot of listeners and viewers, is that we know there are, at any given time, there are lots of clinical trials in a particular disease that are ongoing. Maybe in some diseases fewer than others because the disease is not as common, right? I mean, breast cancer, you're going to have more than myeloma. But these trials are ongoing at the same time, and some of these could be 3, 4 randomized controlled trials at the same time, and they will read at different times. So what happens? You are designing a clinical trial, you're enrolling on that clinical trial, and then another study, randomized controlled trial that is ran in Europe, in Asia, in wherever it is, that reads out, that shows an output or a result that nullifies the control arm that you're working on in your current clinical trial. I mean, you had hypothesized that X is the control arm and you're comparing Y to X. There's a trial going on in Asia that's comparing Z to X, and Z now is superior to X. Well, now you comparing Y to X makes no sense. You need to compare Y to Z. How do you reconcile this? It drives me crazy. It drives a lot of patients crazy, families crazy, and other investigators crazy. And what do you do in situations like this?
Yeah, I think you just described a very common problem that we have faced and I have faced. It goes back to, why are you doing the trial? Okay, so take DARA. So daratumumab Lendex compared to Lendex. We're doing the trial. At the same time, some other company is doing carfilzomib Lendex versus Lendex. Some other company is doing ixazomib Lendex versus Lendex, and another one is doing elotuzumab Lendex versus Lendex. All in relapse myeloma, all in 1 to 3 prior regimens. Okay. You go back to the purpose of the trial. The purpose of each of these trials is really to get a drug that is not yet on the market approved. If Dara Lendex is better than Lendex, it just means that Dara works in myeloma. It doesn't mean that it's the best. It just means that this drug is effective in myeloma. Where it'll work, how it'll work is something that we have to decide later. So the fact that carfilzomib Lendex versus Lendex read out before that doesn't change anything, because that trial showed that carfilzomib works. The ELO trial shows that ELO works. The ixazomib trial shows that ixazomib works. So now we have 4 options for myeloma, and then we can figure out how to use these 4 the best way we can. The fact that one read out doesn't affect the other. And because we literally at that point in time have none of these 4 for any patient. They're all investigational drugs. They all have to get approved. So that's the most common scenario where the drugs are not on the market and we're all each doing a drug trial just to get it on the market. Sometimes what happens is that a trial like, I'll take DARA again. So DARA RD versus RD is going on for frontline. It's already myeloma frontline in the US is VRD. So why are they doing DARA RD versus RD? Or maybe they started off thinking RD is the correct control, but midway through the trial, VRD became standard of care.
Right.
Now they're doing DARA RD versus RD and everybody else is using VRD. How does this even fit in? And those raise challenging questions. And I'm sure what happens with these trials when that happens is literally every big result that comes in where the IRB question is there anything in the literature that affects the risks or benefits to the patients? That all investigators have to answer truthfully. Okay? And most of the time what happens is that the accrual to a trial is either already completed or is going on in a country where whatever is proven the other way even is just simply not a reality. So VRD, for example, has not been approved even in the US even now for frontline. It's just because Vincent's using VRD for frontline doesn't make it the stand, or just because I say it's the standard of care doesn't mean that Poland would say VRD is the standard of care or France would say VRD is the standard of care. Okay, so what happens is all the investigators have to understand, like, if the trial was not there, what would we be doing for our patients in our country? Forget that the US could do VRD. If you could only do MPT or VMP or RD, then you will say, that study that's currently going on is still fine. And I will just let my patients know that, this other trial result came out. So I think, like I told you, in the new drug approval scenario, most of the time it doesn't matter because we need all the four of the all of the drugs, as many as we can. When you're trying to get a new indication, it does matter. And people have to be truthful that, in your country that it actually, did I freeze?
No, so I think what you mentioned is actually pretty interesting because you're alluding to the differences in standard of care between countries.
Yes, you froze, Chaddy. I cannot see you.
Yeah, I froze. I think usually once I freeze, it should start opening. Let me put myself on.
There you go.
Here I'm back. It does do that every so often.
Yeah, you can make your comment again.
Yeah, but I think what you're mentioning is interesting because you're alluding to the differences in standard of care across countries. And across the globe. And I think certainly the standard of care differs based on drug availability. But one of the most common issues that come up is that, okay, well, the trial is being ran globally. And because of that, we are willing to have a control arm that is more aligned across the globe, not a US-based control arm, because we want to make that available to countries outside of the US. But then when the trial reads out and the new regimen is available, that new regimen is not actually available for the people around the globe. It becomes more available to the US because, basically, so the patients outside of the US essentially were enrolled on that trial with the idea to help the drug company get the regimen approved. But when it's approved, the new regimen, these new drugs are not available to these patients in the other countries. They become available in the US because of how big the market is and because the US doesn't have much of a regulation over drug pricing. You know what I'm trying to say?
Absolutely. Absolutely. And this is not a science problem. It is more of a way drug pricing is in the US versus other countries. In the US, a drug can be priced as long as the FDA is approved based on what pharma thinks it's worth. In other countries, pharma has to negotiate with health authorities to determine a fair price for this new drug that just showed value. And what happens is, even though the trial is run across the globe and the drug is successful, the drug almost always first becomes available in the US because they don't have to go through the second step.
Yeah.
So in every country, approval is one of two steps. There's the approval, and then there's the pricing negotiation. So regulatory approval just means that the drug is shown benefit. Then there is the health authorities who negotiate about the value that the drug offers and whether the price that they're proposing is in line with that. And then that can go on for months or years. In the US, there is no second step. Once the drug is approved, it's there. And then as long as it's not like one of these Alzheimer's drugs that we just had,
Yeah.
it's usually reimbursed because it's an oncology drug or an important drug that's for cardiology or something like that. It's reimbursed, and there is no second step that they need to go. So this is not the fault of the PI, or even in companies, just like in big institutions. The medical affairs branch or the clinical development branch, which is doing all the trials, is very different from the economics or the who's marketing the drug and how and where it'll be marketed. The people who are designing and doing the trial don't control if and when negotiations will be successful. So I think while it is true that a lot of these global trials, the approval comes and it benefits US patients mainly for the first year or two, that's a fault of our pricing system, the US pricing system. It's not the fault of pharma or the investigators involved in the randomized trial.
I mean, there is a sense, and I see that sometimes on Twitter, on social media, and elsewhere. And there's actually a couple of papers that were published on that, is that there are many randomized controlled trials that have a control arm that is inferior to what you would normally prescribe in the real world. And that is almost a ploy or gaming the system so we could get the new regimen approved. Do you agree with that statement, or do you think it's not true?
And again, it goes back to what I said, if somebody was not aware of the purpose for the trial, then it's very easy for them to look at the control arm and make that judgment call. In the drugs that I've been involved with, usually it comes from the investigators that if we think bortezomib is active in myeloma, we're going to design a trial that's the quickest way to get bortezomib approved. Because the longer that the trial takes, the longer patients all over the world will wait for the drug that we think will save lives. The more patients it needs, the longer it'll take. The more complex the regimen you propose, the longer it'll take. So you take those factors and you decide, I really have this drug and I really need to show the FDA that it is effective and safe. You could then basically do like a placebo control trial. Forget a weak control arm. You could have a no control arm, like we did with COVID vaccines, just placebo. Just take patients who have no other option and just give them bortezomib versus placebo. Because it'll solve the problem, because without the trial, no one has access to the drug. The drug, as far as you're concerned, a priori is unproven. So you're really not withholding some effective therapy. And so you could do like basically a placebo-controlled trial, just like we did with the Pfizer vaccine or the Moderna vaccine in the midst of a pandemic. We still had to do the phase 3. So even placebo is an acceptable control when you're dealing with a drug that is completely unproven and you're just trying to demonstrate that it's safe and effective. In oncology, particularly when we, as clinicians, like you, for example, if you had 20 patients for whom you gave bortezomib and they all responded very nicely and did well, you didn't have a control arm, you would feel very uncomfortable doing a placebo-controlled trial simply to prove the point. So then you say, okay, you know what? I can't do placebo, so I'll do bortezomib versus dexamethasone. That is what we did. That was the apex trial. That's what got bortezomib approved. So someone could say, why did you do dexamethasone? That's weak. So the purpose of the trial has to be always taken into account. On the other hand, if your trial is a tactical trial where I am trying to decide, I have VRD, I have KRD, which of these two is better for my patient? I have DARA-RD and VRD, which of these two is better for my patient? Should I do CAR T or transplant? There, the control arms both have to be reasonably good so that the control arm has to be correct and good, because you have to make sure, because you're asking an important question that affects management of the patient. So I think a lot of the control arm issues comes from not understanding that the trial purpose was to get a drug on the market in some place in the world.
The there is the designing and taking clinical trial from randomized control trial from inception to completion. What's the average time? How long does it take?
It depends on the trial, Chadi, and what process it is. And again, it's worthwhile going back to what I said. If it's an industry-funded trial, an industry-run trial, it could be very short. You can start a trial within 6 months of the idea, and you could complete a trial within another 6 months to a year. That's how many of these drugs were approved with DARA or carfilzomib or things like that. Because they even in industry, it's not that easy. I mean, the medical affairs or the clinical development branches of these companies have to go through a lot of paperwork and a lot of convincing to their higher ups, because it costs hundreds of millions of dollars to do one of these trials. And so they have to really be sure that this is the right design that they're going to spend the hundreds of millions of dollars on, let alone if the trial flops and it's $1 billion loss. So it's a very high stakes game, but at least they have an internal process. It's rigorous, and then they come up with the idea, and it could be done quickly. On the other hand, if it's a publicly funded trial like the ECOG trial, I'll give you a good example. Sagar Lonial just published the lenalidomide versus observation trial in 2020. I wrote that trial in 2003. Okay.
Wow.
Before the Spanish even started their trial with lenalidomide. And I actually got rejected by CTEP that lenalidomide is not a drug you would want to treat asymptomatic patients with. And this is not the right idea. And then we protested. And finally, we got the trial approved in 2006 or 2007. And then it took 10 plus years to accrue. And then we have to close the accrual and wait several more years to follow up these patients to see what happened. So depending on these trials, it can take a long time. But that's because myeloma is a rare disease. Smoldering myeloma is 10% or 15% of myeloma. So it takes a long time to do this. On the other hand, we have done publicly funded trials like ECOG, where we did the dexamethasone study, that high dose dex versus low dose dex. I think we opened the trial in 2000. I thought of the idea in 2003, and we published it by 2007. So 4 years from idea to opening to accrual to publication.
So, and I won't take a lot of more of your time.
I'm hoping that my calendar times are correct, but very short time is the
Yeah.
basic idea.
So I mean, I think I see sometimes all of this back and forth on social media and Twitter and I mean there are um, there's back and forth always. There's no perfect world that we live in. I think we can always be humble and admit it's never easy to do that. But do you? I mean, is it? Do you have to do a randomized control trial to be critical of randomized control trial?
So, two things I'll tell you. First of all, I am a very ardent proponent of randomized trials, control trials. But I have done 10 and published them, and I've got 3 more that are running right now. So, I have some perspective on what it takes to do one. And it's not that easy. It's very hard to do one of these. Plus, I think the Institute of Medicine had done a study or done a survey, or, for every 100 most important questions, we have funding to do hardly one randomized trial. So we have to really be careful about what we can and cannot do and be realistic about that. So I will say that. And I think that's where I'm coming from. I think it's perfectly fine for somebody who has never done a randomized controlled trial to be harshly critical of a randomized controlled trial, because it's their patients who get affected and they have to treat their patients, and um, they've got good intentions about what is right and what is not right in medicine. And there are a lot of randomized trials that are not done properly, that are not, like we talked about, not right control arm, whatever. So I'm just not having a problem with people criticizing trials. All I'm saying is sometimes you don't want that to be your sole objective.
Yeah.
It helps to try and do these trials to get an understanding for what it takes. I would actually welcome if anyone who has not done a randomized trial wants to um, know what the process is from outside of industry, like not a pharma trial but a publicly funded trial. Just have them email me or direct message me, and I will have them attend our ECOG conference calls. I have right now in ECOG at least 4 or 5 myeloma investigators who have tried for the last 15 years to open one randomized trial, and they've all been vetoed by various steps of the pathway.
Yeah.
Even though they had good ideas, these are bright people. They have agreed to any, like, I come up with a design and you tell me, no, Vincent, don't do it this way, do it this way. And they agreed to it. And then it goes somewhere else, and some 5 other people tell them something and they agree to all of that. And then you get a terse 1-page letter saying, like, your trial was not approved.
I mean, the way I look at things is a little bit, it's simple. I mean, I always say, look, I always think of things similar to a patient coming into your clinic that does not have complete records. And you must make a clinical decision. I mean, you don't always have every piece of information that you desire to decide what you are going to do for this patient in the exam room in front of you. And sometimes you have to pontificate, use your clinical expertise, your judgment, and make a decision. And, similar to that, there are no RCTs for every single clinical question. As much as I would like to see that, it's not there. So, you have to make also clinical judgment in deciding how you're going to treat this patient, despite the lack of RCTs that demonstrate or prove your theory of the best approach. I mean, the 100-to-1 ratio is probably spot on, and some people might argue it's probably less than that. I mean, there are more questions than 100-to-1 to be able to get in RCTs.
Yeah, and I totally agree with you. And I think that's where the judgment call comes in terms of recognizing, number one, that which is a question for which you really need a randomized control trial. And you have a lot of questions and you have to decide which one really needs one. And I have always insisted, for example, if you're going to tell patients with myeloma you don't need a transplant, you need to show a randomized control trial. Or if you're going to tell somebody you need maintenance therapy, you need to show me a randomized control trial with survival benefit. On the other hand, Monday Cilta-cel is going for FDA approval for the second CAR T in myeloma. We have patients who have exhausted their options and they really need some new treatment to prolong their life. I am not going to insist on a randomized controlled trial for the approval of Cilta-cel. So you need to know when you absolutely need one, when you can live without one, and when you can settle for something that's like in between, where you go ahead with it, like DARA, and then wait for the randomized controlled trial to come later. So you do all of that together, because we are living in a world where there's always not just a type 1 error. It's not always like, I don't want to conclude something works when it actually doesn't. You always have to worry, I have cancer patients in front of me who really will die without treatment. And what is the chance I am telling them that this drug doesn't work or has not been proven to work, and I am wrong?
Right.
Where I conclude that this doesn't work or hasn't been proven to work when it actually does, I have to always keep that in mind. And that's a judgment call. And the judgment call comes usually with a real deep understanding of the disease. So I will not be able to tell you what is the best trial for lymphoma or diffuse large B-cell lymphoma or follicular lymphoma or breast cancer, because I lack that depth to know the history of the disease and the situation. It comes with deep expertise, and it comes with a certain amount of experience in terms of doing these studies to know what's feasible and what's not, and how long each will take.
Vincent, before we end up the conversation, this was great. Just like, I feel like it's a primer on RCTs. Anything else that maybe I forgot to ask you that you feel is important, you want to share with listeners? Anything specific that we need to cover I may have failed to discuss?
No, I think we've covered a lot. And I think what I would tell listeners, if they are interested, they can read one particular paper that I'm proud of. That is Philippe Moreau, who's done more randomized trials than me. And I wrote for the Lancet called, I think, Translating Results to Reality for Myeloma in the Lancet. And in that, we outline the problems we face, like, having to choose when we can't do randomized trials, what the process is, and also distinguishing between the various types of trials, the trials that are needed for a new drug approval, the trials that are needed for a new indication for an existing drug, the trials that are needed to outline what is the best course of action for treatment. Should I do maintenance or not? Should I do VRD or KRD? Should I do R-CHOP or R-squared CHOP? And then finally, strategic questions like, should I go for a cure? Should I do targeted therapy, MRD-directed therapy? And those kind of things, how to approach those questions. Your listeners will get more information on by reading that paper. And there are a lot of good references also in there.
And I really think one of the most important things I learned today is don't throw the baby out with the bathwater. Just at least ask the question why the trial was designed, because based on the purpose of the trial, it may be totally logical to continue that study despite the hypothetical possibility that it's no longer valid. Go back to the reason why the trial is there.
Absolutely. And I think something that is really fundamental is, just because we have the Toyota Camry, we don't say we don't need the Honda Accord. The more drugs that we have on the market for a given disease, the better it is from a cost and economic standpoint. There's a lot more competition, and we hope that prices will come down. Now, people can be cynical and say that has not happened. Yeah, maybe it has not happened with various other things. But to be honest, I just saw that, you can get imatinib for $17 a month now. So it will happen with time. And so the fact that I have DARA doesn't mean I don't need carfilzomib. It doesn't mean I don't need elotuzumab. Even if there are two monoclonal antibodies targeting CD38 like DARA and isatuximab, it's good to have both. Sanofi has actually lowered the price of one compared to the other. And so there's reasons why it's okay to have 2 or 3 drugs. So people don't need to get worked up over, I already have these 3 drugs. Why are they studying this new one?
Okay, well, look, thank you so much, Vincent, for your time. Really appreciate. This episode is being taped on the last day of February 2022, and I think it's going to air likely the last day of March 2022. Vincent Rajkumar, thank you so much for coming on Healthcare Unfiltered.
Thanks, Chadi, for having me.